Hello, I am Professor Anowar from Russia University. Today, I would like to show you the Monstera tissue culture. The Monstera are the indoor plants which are usually imported from the Thailand. In Bangladesh, there is no laboratory for producing of Monstera tissue culture plants. So the nurserymen are asking me to produce Monstera plant because the Monstera plants are of high demand in Bangladesh. So I would like to show you a step-by-step guide for uh, Monstera tissue culture so that you guys can produce Monstera plants by yourself. So now let's start Monstera plant tissue cultures and how to prepare the explants and how to sterilize, and also finally we can culture and in the media, and finally next in the after one day of culture, then we can see the results of some contamination or something. So okay, let's start. Monstera eskeleto, and this one is the Monstera borgisiana. Monstera borgisiana. This is variegated plant. You can see here all the leaves are variegated. Here you can see here. So these are two indoor plants. We are going to make the tissue culture. Here you can see some shoot are there. There are 4 shoot here, and here this one has only one very small shoot you can see inside, and another one is this side. You can see another one. So this one have 2 shoot. There are 4 stems with the shoot. So we have to separate the small shoot from the stem, and next we cut into small pieces and we inoculate in the media. Solution: we cut the shoot apical stem, and we just, this is the detergent reagent. It should be cleaned up more than 10 minutes. So just when this time we shake it and then transfer to the another fungicide solution. Okay, thank you. Here we transfer the explant into the fungicide solution, and we incubate with shaking for 10 minutes. These are the explant. So we finally clean, sterilize the this explant with mercury chloride. So now before dipping in the mercury chloride, we have to clean all the mess, all the dirty things adhere with the explant. So I have to clean and then it transferred to the mercury chloride. Okay. 6, 7, 8, 9, 10. So I am shaking the sterilization solution with our explants for the surface sterilization. So we have to sterilize it up to 5 minutes. Just taping, just use the wrap. So wrap is essential to close the lid of the tissue culture vial. So we just wrap the lid here, and that could be safe for transporting cap from one room to another, and sometimes the cap could not fit well. In that case, the wrap is useful. এটা কাটতে পড়ে গেল। Hello, you can see the borgisiana. Borgisiana is still now okay, but another one is the Monstera eskeleto. It has already contaminated. You can see it's already contaminated, and maybe this is bacterial contamination. So the bad luck, it is already contaminated, and another one also contaminated. This one is still now is okay, no contamination, but this one you can see another contamination. Okay, so